Hello and good afternoon, and thanks for attending the webinar regarding CytoViva's Enhanced Darkfield and Hyperspectral Imaging System. Today we're going to focus on the ability to conduct label-free optical observation and hyperspectral characterization of nanomaterials, principally in biological environments. My name's Byron Cheatham, and I'm Senior Vice President at CytoViva, and I'm pleased to provide this information to you today. Before we start talking about the technology and how the technology operates in the nano world, very briefly just want to provide some information regarding CytoViva Inc., which is affiliated with Auburn University, which is a state university in the southern U.S. The company was formed in 2004 specifically to commercialize optical technology designed to advance imaging and analysis at the nanoscale. So let's transition very quickly into the technology and give an overall description of what it is and how it works. At CytoViva, we have developed integrated enhanced dark-field optical microscopy with hyperspectral imaging. So these patented enhanced dark-field optics create a very high signal-to-noise optical image. To give an example very quickly, if you look at the image in the top right here where the cursor is, you can see a live oral cancer cell with internalized gold nanoparticles. So you can see the scatter from the cells and the nanoparticles against a very bright background. And that's inherently how dark field imaging works and the type of image that you're able to see. It's important to note with this image there are no labels. We're seeing the image without any labels or other special sample prep in its natural state. And while this is a very nice qualitative image of the sample that we're illustrating, by integrating hyperspectral imaging onto the optical microscope, the enhanced dark-field optical microscope, we're able to collect a hyperspectral image. The principal difference between a hyperspectral image and an optical image is that in a hyperspectral image, all of the pixels in the image contain the visible near-infrared or shortwave infrared spectral response of that pixel spatial area. So if you look at the graph on the bottom right, you see the spectral response represented in red, which is representative of a pixel from the nanoparticles internalized by the cell. You see the much weaker signal below in yellow, which represents cell structure in the sample. So by having the ability to spatially go to any pixel and understand the spectral response in that pixel spatial area, quite a bit of quantitative information can be derived from the sample so that you understand the spectral characteristics of the sample in its full spatial context. And that's what we're going to cover lots of details on today with regard to the technology and application examples in the nanobio world. So we're going to attempt to cover four things today. Number one is the ability to optically observe nanomaterials without any labels or special sample prep using the enhanced dark-field technology. Number two, observe these nanomaterials in a wide range of biologicals. And these biologicals can be labeled or unlabeled. If we see the image here in the top right, we're looking at multi-wall carbon nanotubes in lung tissue. The multi-wall carbon nanotube has no special sample prep on it. The tissue has a stain on it. And so in this case, we're illustrating the ability to see nanomaterials in a stained environment. Unlike the previous example we just illustrated of gold nanoparticles in an unstained lung tissue. So we're also going to illustrate the ability to spectrally identify and characterize nanomaterials in a wide range of biological environments, and then conduct some fairly extensive analysis, which we'll show some examples of, such as the ability to spectrally map nanomaterials and biologicals in other environments. And here in the bottom right, you see the spectral response of the carbon nanotube against the stained tissue. With regard to this example and another example, we're actually going to illustrate how you can conduct analysis in a hyperspectral image of nanomaterials after we go through some of the overview of the actual technology itself. So before we get into the details of the technology, we want to just very quickly illustrate the footprint of the system. And you see at the core of the system is a standard research-grade optical microscope. The microscope contains the CytoViva enhanced dark field optics in the condenser mount of the microscope. And in the camera mount of the microscope, you see a diffraction grating spectrograph with an integrated camera or charge-coupled device, two of the key elements in the system. You see an automated microscope stage which serves to help move the sample across the field of view of the spectrograph and the detector in a highly specialized way. And this automated stage, along with the detector and spectrograph, are both controlled by very powerful image acquisition and image analysis software, which builds a hyperspectral image of nanoscale materials in what we would refer to as a push broom or pixel row by pixel row fashion. Typically, to capture a hyperspectral image can take seconds or minutes, depending on variables such as the exposure setting of the detector and the field of view that you're capturing, etc. And we'll explain how that works in some more detail as we go through the presentation. So with that, I'm going to provide a little insight on the patented dark field microscopy optics, which allow for the ability to optically see scatter from nanoscale materials and those materials in a wide range of environments. And first, I want to just describe how dark field microscopy works at its very basic level. You see here in the diagram that we're illustrating that the dark field condenser and condenser lens serves to send light toward the sample and the microscope objective at very oblique angles, such that the source illumination from the light source interacts with the sample but doesn't directly enter the objective lens. It has a steep enough approach of angle that it bypasses the objective lens. However, as the light at oblique angles interacts with the sample, scatter from the sample is presented to the microscope objective, such that you're able to see in the image here in the bottom left the scatter from 240 nanometer polystyrene beads against a dark background. And that's the basics of how dark field or oblique angle illumination work. However, standard dark field microscopy optics are not very efficient in the way that they manage the light to the sample and present it to the microscope for observation and image creation. And we want to explain that just a little bit to provide some insight as to how the CytoViva dark field system overcomes these issues. And so as we're looking at the diagram cutaway of the microscope here, you see that light originates from the halogen lamp, the source illumination in the base of the microscope, and travels through the base of the microscope through field lenses and collection lenses, which typically are not very high quality in most even research-grade optical microscopes. After it gets to the field diaphragm at the base of the microscope, this light travels as much as 9 centimeters through the air to the dark field condenser. And this is just a very inefficient way to manage the photons, and so most of the photons are lost before they're ever presented to the dark field system. And because the dark field condenser itself uses an annulus to create a hollow cone of light, there's a very small area for the photons to actually be focused on the dark field condenser. And as a result, this reduces the total amount of photons that are available for dark field imaging, makes it difficult to utilize, and is just not very efficient. So CytoViva's patented dark field optics, which were invented at Auburn University by Dr. Vitaly Voyaginoy and his team in the biophysics lab, they addressed this problem by improving the light efficiency of the optical path for dark field, and also dramatically improving the ability to focus dark field in a highly automated fashion. And we're going to talk about that here. If you remember in the diagram of the microscope we showed a little while ago that light travels through the base of the microscope in the air to the condensing system. With the CytoViva illumination system, the source illumination is actually coupled via liquid light guide directly to a dark field illumination system, thereby having more photons available for the dark field condenser. Now, also, once the light is coupled via liquid light guide, collimating lenses and mirrors are utilized to very precisely take these extra photons that are available and to match them with the geometry of the condenser annulus that we see here above. And by conducting these very simple improvements to the management of the light, we're able to see a very consistent increase in scatter detection, which is observed. This increase has been measured by independent organizations as well as by CytoViva to be as much as 5 to 7x over standard dark field illumination. And so here we want to transition and actually illustrate this difference. And this illustration was done a number of years ago at a core microscopy facility at Dow Chemical Corporation in Midland, Michigan. And first they plated 240 nanometer polystyrene beads on a microscope equipped with a standard dark field condensing system. And we're able to capture the image that you see here on the left, leaving all variables constant except replacing the standard dark field condenser with CytoViva's enhanced dark field illumination system. They were able to capture the image that you see on the right. So from a very qualitative perspective, you're able to easily see this pretty significant increase in signal to noise that's experienced by using the dark field system from CytoViva. So the question that's often asked at this point in the overview is, so what can I expect the detection limits, the scatter detection limits, to be utilizing the CytoViva enhanced dark field system? And what's been published in numerous papers over the years, and what we've experienced here at CytoViva, is that with noble metals and metal oxides and very bright nanomaterials that have very high scatter efficiency, that you're able to detect those materials down to about 10 to 15 nanometers. When we look at soft nanomaterials such as liposomes, etc., typically we're able to see a scatter detection of those materials down to about 50 to 75 nanometers. So signal to noise is really important, not only to be able to observe nanoscale materials optically through the microscope, but if you're going to try to capture a spectral image, high signal to noise is critical. All spectroscopy and good high fidelity spectroscopy requires good signal to noise of the material that you're trying to capture the spectral data from. And so what we want to illustrate here very quickly is what happens with a 100 nanometer particle when we capture a hyperspectral image of that particle and we measure a single pixel spectral response using standard dark field optics. And you see the resulting spectral response signal we get here below with a very low relative amplitude and lots of noise in the signal. Now, if we capture another hyperspectral image of that very exact same nanoparticle and we measure the spectral response of the exact same pixel in this image, and the only difference is replacing the standard dark field optics with CytoViva enhanced dark field, you see a dramatic increase in signal to noise in the relative amplitude. You also see a much cleaner signal that's measured. And just from a qualitative perspective, you can see a dramatic increase in signal to noise from one image to the other as we have them presented here side by side. And we wanted to illustrate that as we transition into hyperspectral imaging and explain at a high level what hyperspectral imaging and how it works consistent with the CytoViva system. So we're showing, again, an image of the optical microscope with the diffraction grating, spectrograph, and the detector integrated onto it, the hyperspectral imaging system. And we're also showing the automated stage here. So this hyperspectral imaging system that can be on the camera mount of the microscope can either be a vis-near infrared or shortwave infrared spectrograph, depending on the applications that you want to use. Most typically, we would say in the nano world that vis-near from 400 to 1,000 nanometers is the spectrograph of choice for measuring engineered nanomaterials in biological environments. One of the things that's important to mention is that there's typically a very narrow slit, about 30 microns, at the base of the spectrograph. And we want to show here in the cutaway illustration here on the right the focusing lenses and the columnating slit represented here in this area. And the scatter from the sample is presented from that 30 micron slit, onto the diffraction grating of spectrograph so that that scatter is broken up into the resonant components of the light and presented on to the camera for collection. So what in fact happens is the microscope automated stage, which is controlled by the image analysis and image capture software, actually push brooms the sample across the field of view of the microscope and the spectrograph to capture all of the spectral data as an RGB, or red, green, blue image, pixel row by pixel row. And that helps to provide you some insight as to how mechanically a hyperspectral system works on the microscope in a push broom fashion, and those components that are at work there. So if we talk about image and data production as a result of that, we're capturing typically the reflectance spectrum from the microscope field of view, as we mentioned earlier, either in the vis-near spectrum or in the swerve spectrum area from 400 to 1,000 or 900 to 1,700 nanometers. And all of this data is captured as a full RGB image, such that what you see in the microscope eyepiece is what you see as a hyperspectral image once the image analysis software has captured this data and presented it for observation. And it's capturing this spectral data in every pixel from 400 to 1,000 nanometers in the Wiener range at very high spectral resolution between 2 and 3 nanometers typically. And as a result of this, detailed quantitative analysis of nanoscale objects in the field of view can be performed quite easily. And so we want to very quickly illustrate the hyperspectral image of the gold nanoparticles, which are green in the image here as a result of the plasmon resonance effect of the gold, not as a result of any labels on the nanoparticles themselves, in a live red blood cell culture. And so we look at the crosshair here on the gold particle in the live red blood cell culture, and we see the spectral response from that single pixel that was queried in that pixel of the image. Again, just to illustrate the ability to conduct a single pixel response. Now, we're going to go through some examples of applications in the nano world for hyperspectral imaging. But after we complete that, we're actually going to open a couple of image files in the hyperspectral image analysis software to better illustrate how they work. But if we talk about examples of analysis that can be conducted at the nanoscale with this instrument, we're going to look at the ability to characterize functional groups or changes in surface chemistry on nanoparticle surfaces. And we're going to illustrate an example here looking at silica gold shell nanoparticles. And all three of these samples that we show in the three different sample images here, 1, 2, and 3, are identical silica gold shell nanoparticles. Except in sample number 2, pegylation was added to the surface of the nanomaterials. And so in this experiment, the design was to see if we can understand the spectral response difference in 3 identical nanomaterials when one of those samples actually has the PEG added. So if we look at sample 1 and sample 3, represented by the green and the red spectrum here in the graph, we see that they produce virtually identical spectrum, which they should because they are virtually identical samples. However, if we look at the spectral response from sample number 2, we see that we have a dramatically different spectral response as a result of adding the PEG to the surface of the nanomaterials, such that we're able to infer that pegylation being added to these nanomaterials as a result of a difference in the spectral response that we measure. And we could capture a spectral library of these two conditions with and without the PEG coating on the nanomaterials, and the system would be able to identify individual nanomaterials in a mixed sample which were and were not functionalized, or in the cell which were or were not functionalized. And so this is one example of the ability to understand at the nanoparticle level functionalization as a result of this technique. Another example we want to show very quickly is the ability to observe nanomaterials that have been internalized by mammalian cell culture. Here on the left, you see a mammalian cell culture that has internalized gold particles, which were targeted with a targeting protein specifically to these mammalian cells. And so if you observe the image on the left with the targeted gold versus the control cells on the right, which don't contain any gold, from just a pure qualitative perspective, you can very easily observe the difference between the gold-exposed cells versus the control cells that were not gold-exposed. And as we go to the next slide and we see a very similar image, different images, but of the same type of sample. We can see the gold nanoparticle targeted cells here on the left. And if we collect reference spectrum of the gold in the cells, and we save that, in this example, as a library spectrum, we're able to take that library spectrum and using an algorithm in the image analysis software called Spectral Angle Mapper, or SAM, we're able to map every pixel in the image that's a match to the library spectrum of the gold particles in the cell. So you see represented in red here every pixel that's a match to the gold in the cells. And as a result of conducting this spectral mapping algorithm, we can also produce a class distribution, which will provide data to illustrate the number and percentage of pixels in the image that spectrally map for the gold in the cells, so that from sample to sample you can get a relative match of the amount of gold, for example, that a cell may have been exposed to versus what was taken up by the cell. It can provide some very helpful information in that regard with class distribution. So with that, we're going to stop the PowerPoint presentation here for just a second, and we're actually going to illustrate hyperspectral image files live to give just a very brief overview of the types of analysis that at a very basic level that you can conduct. And at first I want to draw your attention to the plot that we have here in the center. This is actually the spectral response of the carbon nanotube that we looked at earlier in the PowerPoint in the stained lung tissue. And you can see in the crosshair here we have a pixel that's represented by this curve of the carbon nanotube in the lung tissue. And if we wanted to very quickly try to understand and compare the difference between the carbon nanotube versus the spectral response of the stained lung tissue itself, we see we just moved to a pixel that contains the stained lung tissue. And so you see a couple of immediate differences in the spectral response between the carbon nanotube and the tissue. First, you'll see that the relative amplitude of the signal of the carbon nanotube is dramatically higher, almost 10 times higher than the stained tissue that we collected spectrum of again. And so that should be expected because the properties of the carbon nanotube are much different in terms of their density, et cetera, which would produce this much higher signal response. Also, while they seem to share, if we look here, some characteristics in terms of the main peak, which would be expected as they were both exposed to the stain when the tissue was stained. You see here, though, at about 500 nanometers or so, some fairly significant differences in the spectrum of the carbon nanotube versus the tissue, which are quantitatively different and help to illustrate the difference between, in this case, the stained tissue and the carbon nanotube. So that's one example of comparing spectrum that we wanted to try to illustrate. The second example that we wanted to try to illustrate is the example of gold nanoparticles in a live red blood cell culture. Again, these are unlabeled live red blood cells that had been exposed to gold, and the sample was captured. And so in the zoom image you see here on the bottom with some pixelation, you can actually see where we've zoomed in, and this is showing the zoom area in the broader image, gold particles and live red blood cells. And as I click from pixel to pixel here in this image, you notice in the graph that we have here now in the center that we're collecting spectrum, and that this spectrum seems to be very homogeneous in nature, which it should, because the plasmon resonance effect of the gold in the cells should be highly repeatable, and in fact it is. And so by collecting reference spectrum such as this, one of the things that we illustrated earlier in our example was the ability to collect a spectral library for spectral mapping. So if I wanted to save this family of spectral curves in this plot, I could very easily and quickly save that as a spectral library. And I could select all and save all of those. And now I've actually created a spectral library of gold in a live red blood cell culture. I could then use that spectral library to conduct the spectral mapping algorithm in the software that we illustrated earlier in the PowerPoint presentation. We wanted to show these two very brief examples of image files, hyperspectral image files that have been opened to give you a sense of how they work and operate and their simplicity, and to help you better understand how the technology actually works. And with that, we're going to finish the overview here this afternoon, and wanted to do that, though, by sharing with you just a couple of more applications of use in the nanobio world, specifically as it relates to CytoViva's technology. And we wanted to do that by illustrating a feature and technique that we refer to as dual-mode fluorescence optical imaging, which can be very valuable in the nanobio research world. With dual-mode fluorescence imaging, you're able to, with a special technique that we provide, mix full spectrum excitation light with special excitation light from fluorescent filters, to be able to see what's fluorescent and what's not fluorescent in an image at simultaneously and in real time. And so we want you to look here first at a dark field image of confluent cell structure that contains nanoparticles that have been exposed to the cells. If we look at the image on the right, we see that all of the nucleus, which have a DAPI stain on it, appear in what would be considered a standard epifluorescent type image. If we look at the image in the center, however, when we modulate full spectrum light with the excitation from DAPI using the dual mode fluorescence module that's integrated into the enhanced dark field system, simultaneously and in real time, we're able to see the DAPI fluorescence, we're able to see elements of the cell structure which aren't fluorescent, and we can see the nanomaterials. So that if you're trying to understand in good detail how the nanoparticles, for example, are co-localized with the nucleus, this technique optically will allow you to do that very simply and very quickly. And it's a feature that a number of research groups find quite helpful in their nanobio-related studies. An additional and the final feature that we wanted to illustrate for you today with the CytoViva technology is the ability to do 3D imaging of non-fluorescent and fluorescent elements in cell structures, where we're looking for nanomaterials and potentially their uptake in the cell structure, not just in XY, but also in the Z-axis focal plane. And we want to illustrate this by showing the collage of cell structure that contains gold nanoparticles. And if we start at the top left, and as we move to the right and we move down to the next level, we see that we're going into different focal planes from the top all the way down to the bottom of this cell. And in the middle row of images, where we're in the center area of the cell, you can see some red dots here. And those red dots are actually red dots that represent gold nanoparticles that have been internalized in this cell. And the DAPI nucleus actually outlines the nucleus in this cell, where we're using an algorithm that was developed with this 3D capability known as point of brightest voxel, which serves to identify the nanoparticles based on their brightness intensity in relationship to cell structure. If you'll recall from the earlier hyperspectral imaging that we looked at of the gold particles, and more specifically the carbon nanotubes, where we compared the carbon nanotube spectrum versus the spectrum of tissue, the carbon nanotube had much more intensity in its signal. And the case would also apply here with the gold as well. So by leveraging that information, we're able to identify within the cell areas represented by these very small red dots, the areas where the gold particles have been internalized by the cell. And as we go down further into the cell toward the bottom of the cell in the Z-axis profile, those red areas disappear because we're in a different Z-axis focal plane where the gold particles were not present. And so we can illustrate that now finally with another image, and this is where we would take multiple of those individual two-dimensional Z-slices and reconstruct them as a 3D image, which is illustrated here in this image that you see on the left, where the red would represent the gold nanoparticles in this case. The cell nucleus would be represented as blue, and non-fluorescent cell structure would be represented as gray. To illustrate in this case, which is a different image that we looked at previously, how the gold particles are interacting with the cell in a three-dimensional format. Again, the gold particles are their natural scatter. Part of the cell is fluorescent, in this case the nucleus, and most of the cell structure is not. But with all three of those elements, we're able to illustrate in 3D how the gold is interacting with the cell in this case. So with that, we want to go just to the last slide here very quickly and do just a conclusion of the things that we were able to discuss and learn today. And first is the ability to optically observe nanomaterials without any labels or sample prep, and to observe those nanomaterials as they're interacting with labeled or unlabeled biologicals. We also illustrated how you can see that with fluorescent and non-fluorescent sample elements, and the potential to also observe those in a 3D format. We also illustrated the ability to spectrally identify and characterize nanomaterials, and the ability to spectrally map those nanomaterials in biologicals in a wide range of other environments. So there was a lot of information provided here today about the ability to optically observe and spectrally characterize nanomaterials in specifically biological environments. And so we wanted to conclude by letting you know that if you would like more information about this technology and specifically how it might enhance samples that you work with in your research, that's something that we would be pleased to do. You could send us an email at info@cytoviva.com, and we could have some interaction and dialogue about your samples. And we would be pleased to conduct test imaging of your samples for you here at our imaging lab if you provided us. And also, we have sales partners around the world that can conduct on-site demonstrations of the technique if that's appropriate as well. So please feel free to visit us at our website, www.cytoviva.com, or send us an email at info@cytoviva.com, and we would be pleased to engage you more in this technique and understand how it might help you. And we appreciate your time this afternoon for the webinar, and we hope you enjoyed it. Thank you.